Mr. Nelson.
Anyone listening to that grifter,
You hippo.
petty and immature,
Turn the fan off so you can hear.
narcissistic need to feel important.
Made in the bathtub. There you go. Go ahead, sir.
Uses his natural lack of charisma as a strategic advantage.
You might want to warn your wife.
Petty and immature a senior citizen can be.
I can shut you up.
I should have known better.
There's nothing to cry about, okay? I couldn't believe that guy. He said pine trees and elephants are related.
Oh, I know.
After all, we both have both eukaryotes, yeah. For that matter, elephants and rocks are both affected by gravity. That proves they're related. I mean, what more proof?
But do they fall at the same rate?
Elephants and chickens are related because they both have feathers except for the elephant. I mean, hello, that's proof. Anything becomes proof for their theory in their thinking, okay? It's mind-boggling they could be that dumb.
Standing for Truth doesn't seem to understand the difference or want to understand the difference.
You and SpongeBob need to start a college together.
He bastardizes the ENCODE project, is what he does.
Because the DNA sequences that we possess are largely from living species, we don't yet have a very good analogy for the transitional forms argument. Maybe you could argue with some of the limited data we have. So I'm not going to cover that. But the parallels, again, to the anatomical arguments find an analogy in DNA. Is there genetic evidence that rejects the hypothesis of design? If I'm using design as a competing explanation for the data we just looked at, what about at the genetic level? And of course, Biologos would cite this. If you look at their genetic evidence, they talk about genetic scars, which, again, by its name, implies some sort of error. And I'm not trying to misrepresent them. You'll see some of the detailed arguments are arguments from function. So what is the function of DNA? How much of the human DNA is functional? Some evolutionists might say, well, maybe 10% is functional. That's all you can argue for. Let's think back to the evolutionary arguments for the appendix, for the tailbone or the coccyx, for the evolutionary vestige. What was the problem with those arguments? I'd say there were two problems with those arguments. Number one, they are arguments from silence. Absence of evidence is not necessarily evidence of absence. I'll say it again. Absence of evidence is not necessarily evidence of absence. It might be, but it might not be. So it's already on a shaky foundation. What makes it even more shaky, and I think a second reason these arguments had faced some severe challenges, is the absence of evidence was not derived from decades of experimentation that failed to identify positive evidence for function. The absence of evidence arose from the absence of experimentation itself. So then no surprise, as experiments progress, that evidence of absence disappears and we find evidence for function. So when evolutionists might say there's little evidence for function in the human genome, the first question to ask, of course, is, well, what experiments have been done? The gold standard is genetic knockout. Fortunately, that's still considered unethical in humans. We shouldn't be guinea pigs where someone goes into you and tries to knock out your DNA just for knowledge's sake. But there are individuals around the planet that have, naturally, because of mutations, because I would say because of the fall, they're missing sections of DNA, or somehow some of their genes have been rendered non-functional. Now, only 6%, I think this is an Icelandic study, only 6% of the genes in the human genome have this sort of individual representation, which means the vast majority of our genes have yet to be tested by the gold standard for function. And since genes represent less than 5% of our total DNA, you can easily see then that the vast majority of our DNA sequence has yet to undergo an experimental manipulation. There's been biochemical studies, you may have heard of the ENCODE project, that they said there was evidence for function in 80% of the genome, and it was criticized as being premature. It is premature. It's only biochemistry. It's not genetics, and they're only looking at a small subset of cell types. There's all sorts of developmental windows they did not have access to. And again, you're running into ethical issues here. What are you going to do? How are you going to access a baby in the womb and test the function of DNA in those tissues? But I think we have the same problem as we do in anatomy and physiology. Absence of evidence is not necessarily evidence of absence, and the absence of evidence is from absence of experimentation. To me, what the ENCODE project suggests is a trajectory that is pointing towards increasing levels of function. We won't know if that trajectory holds up or not, but it looks to me like the evidence is pointing towards high levels of function in human DNA. And again, that's what it looks like to me. That's a hypothesis that's testable. And I anticipate then that as that level, the evidence for function goes up, the arguments then will have to necessarily change. And let me just pick one example real fast for sake of time. This is the ENCODE project. I'll skip that again. There's a study of pseudogenes. So you can see in the name pseudogene, even in the name itself, it sounds like this was a gene that once was and no longer is. It used to be a gene. It's broken. Well, the ENCODE project studied basically every single letter in the human DNA sequence. And since there's pseudogenes in the human DNA sequence, it's no surprise these were studied as well. Well, similar experiments have been done in other species, including worms and fruit flies. And the pseudogenes in these species have also been investigated biochemically for function. And not surprisingly, the number of human pseudogenes that have at least preliminary biochemical evidence for function mirrors what you see across the entire genome, about 80%. And similar numbers, 70-ish% in worms and flies, also hold true. Again, this is not genetic knockout experiments. It's not the gold standard. But it does set a trajectory. I don't think the evolutionists anticipated this sort of thing.
The fact that, phylogenetic systematics, as you're talking about here, it's mostly based on the concept of outdated junk DNA. So since, functions are only beginning to be found, say during the last decade, meaning we actually understand less than 5% of the DNA language, the trajectory, and I will also say that a lot of the data is preliminary, but the trajectory is most important. It strongly suggests that the vast majority of DNA will be functional. Now, why that does not bode well for phylogenetic systematics is because this takes away the primary assumption of evolutionary phylogenetics, which is actually based on mutations for one and pseudogenes, right? Genetic markers. But if the vast majority of DNA is functional, as we're seeing now, Godless Galaxy, it now becomes a common design argument. And what's nice and revealing is that creationists have always predicted that the vast majority of DNA is functional. So this functionality is certainly a problem and inconsistency for phylogenetic systematics. What do you think?
So you're saying that because we do not completely understand each function of DNA, that's like one of the reasons why we can't use phylogenetics?
Well, because a lot of these, phylogenetic trees, they're based on genetic markers that are assumed to be, mutation related, or let's use pseudogenes, for example, plagiarized mistakes or what they would call them, right? And if they really are plagiarized mistakes, that would certainly be strong evidence for universal common ancestry. But in regards to the ENCODE project, they found out that at least 80% of the non-coding DNA was actually transcribed into RNA, right? So that actually suggests function, but it's also been found that these non-coding regions, and most of our genome is actually non-protein coding, I'm sure you're aware of that, but it's now known that these regions of genomes correlate with biological complexity even better than the protein coding regions. So that actually suggests that these non-coding regions, Godless Galaxy, they carry significant information, say for regulatory activity. But a big thing as well is ENCODE. They didn't look at the portions of the genome involved primarily and exclusively in development. So that type of DNA would only have function then. So that means the trajectory of discovery favors genome-wide functionality very strongly. Probably, there's certainly a lot more function in the genome than evolutionists formally assumed. But the point is if these genetic markers are actually functional DNA differences, they can't be used as plagiarized mistakes to build these phylogenetic trees. That's the point. You know what I mean? That's the point. So now these trees just remain in the realm of hypotheses. Right now, that's why the battle, rages on. Did ENCODE really detect function, or is it just spurious transcription? But if it really is function, once again, genetics points to common design. Like, do you understand what I just said there?
Sort of. So you're saying because these markers, they will claim to be mistakes or whatever, but that's a way to disprove evolution?
Well, because a prediction based on, pre-existing heterozygosity would be that the non-coding regions or functional. Okay, but evolution says that there will be much non-functionality. So they use a lot of those, say, pseudogenes, which are actually in those areas, as genetic markers to build their trees. But like I said, in regards to the ENCODE project, these results have suggested that at least 80% of the human genome is functional. But as I said, is the results are preliminary. And I've talked to scientists on this. I wouldn't want to straw man the.
Matt, I listened to Stand Up for Truth. I know his arguments. He doesn't understand the problem with heterozygosity.
One last question.
So you believe in Noah's Ark, and there is a certain number of kinds on Noah's Ark. I'd guess probably around 7,000, just to throw a number out. And right now, there's currently about 6.5 million species of animals that live on land. That means that approximately 4 species would have to be created every single day, which by species, I just mean about like several 100,000 differences in the genetic code. So is that an accurate representation of your theory, that you believe that there is a certain number of kinds on the Ark, like 7,000, and there's currently about 6.5 million estimated species? So that would mean that
are you asking me how can you get that amount of species in 4,000 years?
Right, because there'd have to be at least like 10,000 differences per day.
Yeah, I actually, that was a big portion of my opening with the created heterozygosity hypothesis. If you've got these heterozygous ancestors that God encoded with these pre-existing functional DNA differences, because we know there's millions and millions of DNA differences, nuclear DNA differences that separate species and families. So if those are encoded by God at the start of their existence, an almost limitless variety of combinations of chromosomes, genes, and traits are actually possible. So when you look to that model, we don't just look to mutations there, Tom. We look to recombination, gene conversion. They're going to provide many new varieties of these combinations of traits that I'm talking about quickly, since the differences are already built in. So it makes testable, falsifiable predictions that I've talked about earlier.
My question was a little bit different. I was asking because in order to account for those species, you would need to have 4 new species every day. And a new species, I'm simply just defining as something that has hundreds of thousands of genetic differences, which means in order to account for these new species, you need hundreds of thousands of differences every day. Your theory seems to have a much greater evolution than ours.
I just don't think you understand what I mean by built-in genetic differences. There's millions of DNA differences in the nuclear DNA. So if these were built in at creation, created heterozygosity hypothesis, obviously through recombination and gene conversion, these new traits, new species would come about quickly, especially post-creation, immediate post-creation, post-flood.
I think you're saying that it's pre-programmed, that the species are pre-programmed into the DNA?
The genetic diversity is pre-programmed. Actually, there's a paper out. I would recommend you look at it. It's a fulfilled prediction from, based on the created heterozygosity hypothesis. It's about finches, because we predict shifts from heterozygosity to homozygosity, right? Heterozygous DNA sites to more homozygous DNA sites would produce new species quickly, even in observed time. So we've seen just recently, observed time, new finch species came about through shifts in heterozygosity to homozygosity. So because we've just studied speciation and speciation events recently, the more that we do study, that's another testable prediction. There's going to be more observed cases like these finches, many more. But the thing is, it has slowed down since the flood. So there's certain rates, mutation rates, predictions have been made. I would just tell people to read Replacing Darwin, very, very complex technical book, but it's all there. It's very fascinating.
In Psalm chapter 62, it says, "They only consult to cast him down from his excellency. They delight in lies." And in this series, we are covering some of the lies in the textbooks. Now, I don't want to hurt anybody's feelings. I really don't, okay? I'm trying to help. But you were lied to. There is no Easter bunny, okay? I'm sorry, there isn't an Easter bunny. I'm sorry, you were lied to. There is no Santa Claus. And reindeers can't fly. They lied to you, okay? And you were lied to. Superman can't fly either, all right? And you were lied to. You can't fly either just by putting on your underwear outside of your pants, okay? That will not make you fly, okay? And Batman can't fly either just by putting his underwear over his pants and putting his blankie on his shoulders. That doesn't make him fly, folks. It's called imagination, SpongeBob stuff. And there never was a race between the tortoise and the hare. It's a story, a made-up story. You were lied to. There never was a purple hairy dragon that could make himself invisible. Pete's dragon it's a lie. It didn't happen. Most of the fairy tales, I'm sorry, There never was a purple genie in a bottle that sounded like Robin Williams either. It's just a lie. Now, put your mommy's red towel back in the drawer and take the red underwear off and your red socks off before your neighbors see you. You are a big boy now, and it's time to stop pretending. Okay? There are some lies in the textbooks that we're going to cover, and it's time to stop pretending that these things are true. They're not. Grand Canyon was not formed slowly over millions of years. That's the first lie we covered. There is no fossil record. That's a lie we covered. There are lots of fossils, but it's not a record. It's a bunch of bones in the dirt. You were lied to. The fossils do not show any evolution at all. None. How could they? All you know is it died. You found a bone in the dirt. You know it died. You don't know it had any children. In an honest court of law, not one single fossil would last 1/2 second as evidence for anything other than it died. Quit using that as evidence for evolution. It's dumb. Adding time won't help. I'm sorry, you were lied to. Someone told you that over billions of years the frog can turn to the prince. They lied to you, okay? Stop. Wake up. You're a big boy now. Face the facts. It's a lie. Adding energy won't help. It's not going to cure the problem that evolution has. The present is not the key to the past. Catastrophes change things rapidly. The Bible says they're ignorant of the flood. That flood rearranged the real estate big time. Yeah, really big time. You were lied to. There is no geologic column. It doesn't exist. The whole thing is imagination. It's a bunch of fossils somebody stuck in order to try to make their theory true. You were lied to. You're a big boy now. Get the blankie off your back and look at the facts. The flood did this, not millions of years of evolution. They say, one thing we've learned by studying fossils is organisms are constantly changing or evolving. That you can't be more stupid than that. You can't study fossils and see that organisms are changing or evolving. None of those fossils talk. None of them have a date stamped on them. None of them have a tag with them telling anything about them. You're putting your interpretation on them. They're not showing any change over history. You're making this up, okay? Quit imagining. There is no Santa Claus. There is no Easter Bunny, and there are no fossils that show evidence of evolution. Wake up, okay? Scientists use index fossils to determine the age of rock layers. This is flat dumb, okay? Index fossils do not determine the age of anything. The whole thing is a lie. There are no index fossils. We covered that a couple nights ago. Nobody went to jail for teaching evolution. This is a lie. And there are no laws against teaching creation or the Bible. This is simply a lie. You can teach it if you want. The lobe-finned fish like the coelacanth is not an index fossil. That's a lie. They're still alive. And stop pretending your grandpa was an ape-like creature. It's time to face the truth like a big boy. There is a God, and it's not you. Stop pretending your grandpa was a protozoa. You're embarrassing me in front of the neighbors, okay? Stop. Stop pretending. It didn't happen. None of this happened. You drew a bunch of lines on paper, and that is somehow evidence. Think about that for 5 minutes. You got lied to by somebody. You came from soup, and the soup came from the rocks that got rained on and dissolved. Yes, you teach you came from a rock. Stop lying and say you don't believe that. Admit it, and show some respect for heaven's sake. Why do you walk on them on your sidewalk? This is your ancestor. Don't you have any respect whatsoever? You mix them up with cement and flatten them out and drive on them with your car. How disrespectful can you get to your ancestors? Gee.
Hey, Aaron, just an FYI, the paper Standing for Truth uses all of this for its BS. It was written by a young earth creationist named John Sanford. It's hilarious to read, published on Biomed.
Sanford's genomic entropy theory. It's crap.
Yes.
Hello, Dr. Stanford. First of all, I just want to thank you for having your talk. I'm involved in a fairly significant group, and your work comes up quite often. My question is a bit of a two-parter. First, you stated that there was 700 billion new mutations per generation, correct? At a genome size of 3 billion, wouldn't we, for simplicity's sake, sticking with point mutations, have flipped every single mutation possible multiple times over?
Yes. Yes,
Okay.
basically every possible mutation that could happen in the genome has happened during our lifetime.
Great. And then you stated that most mutations are in the non-selection range. Wouldn't at some point you reach an equilibrium where the deleterious mutations flip and return to the previous state?
So flipping of back mutations are really rare. So you'd have your weight. Basically, the new mutations are flooding in. Back mutations are exceedingly rare events, and so it'd be just as if there was no back. There is back mutation, but it wouldn't stem the tide.
Thank you.
I wonder whether I got that right. You're arguing that the baby differs from both parents by 100 nucleotides.
Yeah, 50 from each parent.
What is actually the experimental evidence for that? We do next generation sequencing and everything, so one could test that today. Is there evidence for that?
Yes, so I think most of the data is coming from parent-child trios, and so they're actually doing sequencing of parent and child, and so they actually can count the mutations. There are parts of the genome where they're not countable, like in tandem repeats and things, so they make some adjustment for that. But the 100 mutations per person per generation now is widely being circulated as the actual mutation rate. What fraction is neutral is debatable. Yes?
So have you ever considered a situation with some invertebrate species like horseshoe crabs that have been around for a long time into the geological record? How does a species like that continue to exist?
It's really a great question. Kondrashov actually talks about that. He said these lineages should have a lifespan limit. And so one author I didn't include was Dr. Fred Hoyle, a famous physicist. He actually was so interested in this that he spent a few years of his life looking at the mathematics of evolution. And he concluded DNA is degrading, and he said any given piece of DNA has a limited shelf life, basically. And so he envisioned aliens coming and reseeding the planet periodically to make up for all the degenerated genome. So, so really, it's hard to imagine deep time lineages surviving unchanged like the horseshoe crab. It's really a conceptual hurdle. And it's, I think, should be acknowledged widely that how, if current, so one really interesting thing, jumping to the virus issue, is we watched the H1N1 virus go from a red-hot pandemic to a whimper, to an extinction event in 90 years. There's a paper recently published that say that all RNA viruses of all different families appear to be evolutionarily young, meaning tens of thousands of years old. Whereas you'd think that viruses would date back to millions or billions of years ago. And so it's really interesting. The time element is something I don't want to deal with. I just want to deal with the present.
I'm wondering what your starting point is, because you're talking about genetic degeneration in humans. What is the point where you're starting from? Like, what's the point where, I'm just curious, because you have a continuity of humans coming from some ancestor that we wouldn't consider human. What is the point that you're starting from which
So the starting point, that's a good question. Basically, if we start with a population with 0 mutations, there is a burn-in time where it has to reach equilibrium, where it takes time for enough genetic diversity to build up in a 0 population, a 0 mutation population. And so there is a time where it's unrealistic, the degeneration rate is unrealistically high. And so we acknowledge that, but we could start anywhere. So we could start, let's suppose that all of the human beings in the world disappeared except the people in this room. We could start and say, okay, the average fitness in this room is 1. And then we're going to monitor fitness, not as reproduction, because that's a circular argument. We monitor fitness by function, like IQ or physical strength or things like, or longevity. those things are a better way to measure it. But it's a great question, and it is a part of the formulation. We do start with mutations with zero. We start our populations with 0, and so that's a factor. But we can restart a population once you've reached an equilibrium, a burn-in point, and we still see decline.
Thank you for a fascinating talk. I was just wondering, the mutational load is, as you say, is very high, but there's a lot of redundancy in the function of the human genome, and I'm just wondering if anybody has looked at it from that perspective, that there are similar functions. So maybe a particular mutation may take out one key gene, but there are redundant systems that can actually take over that function.
Yes, so that gets in a little bit into the philosophy element, because it looks to me, and a lot of people, like the genome is designed to be stabilized by things like diploidy, or things like gene family backups, or other types of redundancy. So redundancy does, in modern design, engineering design, we look at redundancy as a really good thing, and it is a good thing for our genome. And it's interesting because how do you evolve processes that only have long-term effects? Normally, selection can only act upon things are short. Beneficial mutations have normally a short-term vision of where they're going, so to speak. Yeah.
It's also kind of a two-part question. The first thing is about formulations from Fisher, right, and the early on population geneticists. So those are incredibly useful and formal, but they're also extremely simplistic. They assume, as you're assuming still, like 100 years later, that fitness is just one-dimensional feature, and that the function of selection is to increase fitness, when it's probably this multidimensional feature, and that the function of selection would be to maintain the ability to adapt, and not increase fitness. That's just the construct that was used mathematically to formalize this.
Right.
And this will lead to the second part, which is the equation should be, as was mentioned, for other species, correct? So for bacteria, for instance, and this has been studied, so this could be looked at. If your predictions are right, this should be observed in bacterial populations. They should collapse after a little bit of measurable amount of time in the lab, because they go through a lot of generations, and you can measure more accurately the distribution of mutations and the number of generations, and even sequence everything and see the predictions. But that doesn't seem to happen.
Okay, those are all really good points. Let's see if I can remember what they were. The first one was oftentimes it's not about gain in fitness, but adaptation.
Right.
I totally agree. Adaptive mutations are real. We can document them. They have value, but they don't necessarily increase net information. So I look at adaptation mutations mostly as fine-tuning systems. And so, in a sense, adaptations let a species remain the species because the species can accommodate changing environments. The second issue you brought up was multidimensional. I totally agree that the typical population genetics formulas are crazily oversimplified. The reason we went to numerical simulation is we call our numerical simulations comprehensive, because we try to consider all the variables, and rather than just like a lot of in the past, numerical simulations only simulate some part of evolutionary process, or like, let's say, the rate of drift or the rate of and they often have simplifying assumptions like truncation selection or no environmental variance, things like that. So to do good numerical simulations, you have to consider like 30 parameters, and they're all adjustable. But you can look at a wide range of possibilities, but you realize that you can't reduce this to a formula. And so I agree with that. Your last point was?
Yeah, exactly that point. If you can't reduce it to a formula, you can't model realistic systems. And then when you look at systems like bacteria, where this is not happening, for instance, so I was talking about bacteria. which
Right.
relates
Okay,
to
so
that
the
point.
bacteria is also a good question. So I recently read a paper about reductive evolution in bacteria, and the long-term E. coli experiment also reflects this, is bacteria rapidly adapt to a new medium or a new environment. And that's really interesting. It's an adaptation. They're not gaining any new function. All those experiments usually actually involve reductive evolution. For example, all of the beneficial mutations in the long-term Lenski experiment were loss of function. Genes were deleted, genes were silenced, or genes were downregulated. So in one case, a promoter, which is normally regulated, became an unregulated promoter. But all that is adaptive fine-tuning. There was no new information being created. And in fact, technically, as the bacteria jettisons unnecessary genes for that environment, they are actually painting themselves into a corner. They won't be able to survive in any other environment. So they become handicapped in a sense. So there's another paper on reductive evolution in bacteria where they say, they were looking at a specific bacteria. They said 1/3 of all random deletions increased fitness. It has to do with getting rid of any genes that aren't necessary for the moment. It's very short term. The bacteria, it works for them short term, but then when they need those genes that they've jettisoned, they don't have them. So that's even that, in my opinion, that type of adaptation is entropic.
Thank you.
Yeah.
Thank you for such a provocative talk where you give us the thesis that according to current biological paradigms, we don't exist. But my question is so much of that thesis hinges on a simple piece of evidence that we don't have a normal distribution of deleterious and beneficial mutations. And my question really is, how can we empirically test this when we can only look to the past and present and we actually don't know the future of mutations?
A number of geneticists have tried to quantitate the actual distribution of mutations. It's actually hard to quantify the near neutrals because they're so neutral that you can't see the effect. And so there is a certain amount of assumption there. Let's see, lost my train of thought. Your question was, how can we know the distribution of mutations?
Sure.
And so Lenski's experiment is really interesting because they were periodically sequencing the population as it changed, they could actually determine exactly, and they could determine when mutations arose. They could see which mutations were beneficial. Only a tiny fraction were beneficial, and they were all reductive. So that's a quantitative thing. Also, we did a research on the influenza virus. So Rob Carter and I, turns out that over the last 70 years or so, medical people have been freezing influenza samples. And then all those samples through history were sequenced. So we have this historical sequence, kind of like the Lenski situation, but with influenza. And what we see is that, actually, let me just show a slide here. This is our numerical simulation. Mutation goes up perfectly linearly, and fitness goes down with a fitness decline curve. And this is the influenza study that we did. Rob gathered sequence data going back to the 30s, and what he sees is a strict linear accumulation, just in the same way. So basically, it's clock-like. 10% of the H1N1 influenza virus mutated. And so this is genetic entropy at work, and it's supported by other people who have been following H1N1's virulence, and they show that the decline since again records were being kept, or actually since the 1918 influenza, is that the virulence declined in the same type of way that we see in our simulations, to the point where in 2009, that human version of H1N1, there's still H1N1 circulating that's swine flu, but H1N1 went extinct in 2009. So that's clearly indicating that this is not neutral or adaptive. This is genetic entropy at work in a biological system where we can measure what's going on. There were mutations, by the way, there was some adaptation. There were some beneficial mutations happening, but it wasn't enough to stop extinction. And these two other curves were two other influenza outbreaks, and they similarly underwent entropic decay. They didn't go extinct yet, but they're clearly headed that way. So we do have some biological data.
Okay.
Well,
thank you.
It's true that it's largely theoretical, but there's now growing biological data.
Thank you.
Yeah. So I really appreciate you showing up, and I know I've taken too much time. I have a few books that I'm happy to share with you, one genetic entropy and one on contested bones. If you think you're going to read it, you're welcome to a copy. The BINP, Biological Information, New Perspectives, that proceedings is in the NIH library, and you can, but you don't want to read that proceedings, it would take you a few months. It's 25 really long papers, very technical, on all the different dimensions to biological information. But there's like a synopsis you can read in a few hours, which is freely available at BINP.org. It stands for Biological Information New Perspectives.org. And there you can download the synopsis for free. So if that interests you, that's available. But thank you so much for your patience.
Matt, I listened to Stand for Truth. I know his arguments. Kumara's theory to resolve that dilemma. He doesn't understand any of this.
Now, a lot of evolutionists will say, oh, genetic entropy has been refuted, but it has not. And when you look at rebuttals from evolutionists, whether it's Apologia, whether it's a certain blog on the internet, letters to creationists, when fighting this truth, what their rebuttal will be, they'll speak of the obvious. Beneficials do happen. Selection does happen. Adaptation does happen. No one's disagreeing with this. Anybody with a basic understanding of genetics understands and knows these things. The argument of genetic entropy, genome degradation, only begins after acknowledging these obvious things. So let's look at some of the objections to genetic entropy. A lot of time they'll spend, and I know Jackson, we spent a lot of time saying that John Sanford misrepresented the data, misrepresented Camorra, but this is a rabbit trail. If you're looking at the slide here, John Sanford shows a larger zone of no selection than does Camorra because Camorra's analysis of the problem was oversimplified. The crucial issue is about defining the correct distribution of mutation effects. The vast majority of beneficials will be unselectable. Anybody can easily see that most mutations fall within the no selection zone and that almost all of them are deleterious. So even with strong selection, the entire zone can only undergo degeneration. Now, they'll appeal to beneficial mutations. That's what it comes down to. But due to the influx of deleterious mutations, the rare occurrence of beneficial mutations cannot compensate for the information loss. Beneficial mutations are very rare. Even evolutionists agree to this. And near neutral mutations are continuously and consistently deleterious, just as you would expect with typographical errors in a text. Most beneficial mutations are too subtle to be selected for by natural selection.